Good afternoon, everybody. Thanks for coming. Today, I will go on with the tools that and methods that we were discussing to build a dynamical model to study dynamical processes. And this is so far what we saw in the previous sessions. And if you remember, I finished the last session with an example of doing a qualitative estimation of parameters. It means that instead of quantifying the values of parameters using a model to fit some data, we develop a model to qualitatively explain some phenomenon that we can observe, for example, in hepatitis C infected patients. So today, I will be more focused on. So today, I will focus on more quantitative modeling, and I will show an example where we use the simulated annealing to fit parameters. In the session that Vince gave yesterday, he explained the simulated annealing very well, very deeply. And today I will just go through this optimization method fast, because my idea is to focus at the other part of the session on macroevolutionary algorithms. These algorithms are not so usual in the literature. So when you look for data fitting, apart from the deterministic models, methods, the Levender-Marquardt or gradient descent, you usually find genetic algorithms. Macroevolutionary algorithms, as you will see, are close to the genetic algorithms, but let's say the scale of evolution that the system undergoes on the landscape is different, because the genetic algorithm focuses on what we call microevolution, but this one is focused on macroevolution. So we have like macro extinctions, and I will give you a reference that it's very well explained, and there is theory also behind this method. And usually, well, in the reference, you will find that depending on the landscape or depending on the problem, macroevolutionary algorithms perform better than genetic algorithms. But it doesn't mean that they are better. It depends a little bit on the data that we have. Okay, so this is so in order to illustrate the quantitative modeling that I will be discussing in the next few minutes, I will give an example of a research line that I have been working since 2010, more or less, up to 2 years ago. Now we are not focusing on the specific case. And this is a very important question, that is how viruses amplify the genomes inside the cell. This could seem a trivial question, and as I will show you, there are early works that try to know how these genomes, how the genetic code of the viruses are amplified or are replicated inside the cell. These modes of replication, what we call the replication modes, as you will imagine, have a very big impact on the evolution of the virus, and we will see some examples. So this is a video that I got from YouTube, and it's about it's a simple animation of how a positive sense RNA virus infects a cell. So just for you to know, we have a lot of different viruses that are usually classified in the multimer classification. They are usually classified by the type of genome. So we have viruses with a single-stranded RNA. So it means that we have a virus, let's say, that carries a positive sense RNA, single genome. Okay, and then this virus can infect a host cell. And this is the video I will show you. But then we have other types of viruses. We have retroviruses, like, so for example, this could be SARS-CoV-2, very familiar to everybody, hepatitis C virus, Zika, for example. These are called single-stranded positive sense RNA. But then we have other ones that, for example, carry negative sense RNA. These are also called genomic RNAs. Okay. We'll explain the differences between these. This is, for example, Ebola virus, which is a very dangerous virus because it's very virulent. Then we have also retroviruses that are viruses that infect the cell, and they have a retropolymerase, retrotranscriptase, and they put the RNA, they convert it to DNA and integrate in the host cell. This is, for example, HIV. Then we have also viruses with single-stranded DNAs, single strand of DNA, and double-stranded DNA. So we have a lot of different types of viruses. So here you will see this is a bacteriophage. This is a cartoon. Macroevolutionary is a virus that infects bacteria. And they are viruses that can be RNA viruses or DNA viruses. So here what we have is that the bacteriophage by means of a receptor. So this is the single-stranded RNA. By means of a receptor of the bacteria, recognizes this, it's called sex pilus, and so it means that they are specific. Then this genomic RNA that is from the virus is recognized by the cell as its RNA, as an internal RNA. And then the cell translates the RNA to synthesize the viral protein. But this is the cell machinery that synthesizes the RNA polymerase. The RNA polymerase is depending on the virus, it's a set of proteins that will make copies of the viral genome. Okay? So then, from this polymerase, we will obtain the negative strand, which is called antigenomic. And from the negative, we will try obtain more positive strands. And here you can see that the polymerase is attached to the negative or antigenomic strand, and a lot of positive sense genomes are produced. And then the genomes are encapsulated because the cell also produces the structural proteins that have formed the capsid, and then they are released to the liver in order to progress with infection. So the difference between this one and this one is that, as I said, when the positive sense single-stranded RNA is injected into the cell thinks that this is a messenger RNA of the cell. And then the ribosomes, the ribosomes of the cell synthesize the genome of the virus. The negative sense RNA is different. Because if we have, imagine that we have this, if we have the negative sense genome, the cell will not recognize it as an own RNA. Then this type of viruses need to encapsulate also the polymerase in order to be effective, because here we will have the negative and the viral polymerase, and then we will have the positive, and then all the process will start. So this is a schematic diagram of the infection cycle, very simple. We have the introduction of the messenger RNA, of the viral genome, which is translated because the cell processes it as if it was its own RNA. And then we have, usually, depending on the virus, for example, Hepatitis C, I think it has like 9,000 bases. It's 9 kilobases. But SARS-CoV-2 is 30 kilobases, and Paul knows it very well. It's very long. So usually what we have in small genomes is that we have overlapping genes. And what we have, it's like a protein that is catalytic and self-cleaves itself. So we obtain different proteins. These proteins, these viral proteins that are synthesized by the cell ribosomes, can be two types of proteins: the polymerase or the structural proteins. So the polymerase is the one making the copies of the genome. Okay, this is what we call transcription. And we are also called non-structural viral proteins. And the structural viral proteins are those proteins that are used to build the capsid, and then the virus is released. So this is very simple, but now we will focus on this process here. So these are some articles that we have published concerning this dynamics of this mode of replication, and today I will focus on two. So this is a diagram where we show different replication modes or different replication mechanisms. So we have several options, but we have like two extreme possibilities. The first one is that the initial viral genome is in the cell. Then after the synthesis of the polymerase, we have the negative genome, which is the antigenomic genome, and then the genome is used as a template. So the replicase attaches to a negative strand and starts producing positive sense strands. And this is what we call, or what is called, stamping machine replication. It's like a photocopy machine. Then, in the other extreme, we have that one positive gives a negative, and then a negatives can give positives, positives can be negatives, and negatives can give positives. So each genome has like the same probability or the same rate of production. And this is called geometric replication. Of course, these are two extremes of a continuous because we may have mixed modes. Okay, so this is something that was not known a few years ago, but there are some works that provide evidence of the type of mechanism. One important thing, we know that viruses make a lot of mistakes. They have large mutation rates, very large mutation rates. And you can imagine that it is not the same to have this mechanism than this one. Why? Which mechanism will accumulate less mutations? Any guess? Which one will accumulate less mutations? Stamping machine or geometric? This one, right? Because we are copying all the time the same template. If we are copying templates that already have mutations, this will amplify. The first works that are very old, are from the 60s, 70s, were trying to estimate this thing, but indirectly. How? With the distribution of mutants. If we have a stamping machine replication, the distribution of mutants or a number of mutants follows some distribution that has like this expression where mu is mutation. But if we have geometric, this is for the stamping machine. But if we have geometric, we have a more complex distribution of mutants that follows something like this, where mu is mutation and n is the number of replication cycles. And this is called Woodian-Elbrook. Woodian-Elbrook distribution. Of course, if we quantify the distribution of mutants, we can obtain deviations from a Poisson, and it will indicate that the process is not completely stamping machine. So we'll have some contribution of geometric. So these are the works. These are from the 60s and even earlier from the 50s, the famous works by Lydia. And what they did is to, this is when the sequencing of genomes was working. And what they did is to compute the fraction of mutants produced in infected cells. These are some bacteriophages. Beta is a phage that has an RNA, a single-stranded RNA. If I'm correct, this one has a single-stranded DNA. So what they observed is that the fraction of mutant genomes produced per infected cells for these two bacteriophages follow this distribution. Later, there is a very recent work that follow a similar approach as we did, but this one, reference 3, which is 51, for bacteriophage T2 and poliovirus, they identified this distribution. So far, reference 1, 2, and 3, which you know are quite old, this is not very old, but the other ones are quite old, they were estimating these processes and directly from the expected distribution of mutants. Then, for bacteriophage 5, 6, and terminal mosaic virus, that is the example I will explain to you, intermediate modes of replication were found. So, in the sense that you did not obtain a perfect Poisson, but you obtained like a slight deviation from a Poisson. Okay, so when I did my postdoc at Santiago Elena, they were interested in studying this phenomenon dynamically, not from the expected distributions that one may expect. And they perform an experiment to estimate the replication mode directly from the production of RNAs, positive and negative sense RNAs. You can imagine that when you are doing an experiment like this, you need to try to do a simple experiment able to reproduce what you want to study. So here there is a lot of microbiology and biology, but the thing is quite easy. So what they did is they took a plant virus, it's a terminal mosaic virus. They introduced the genome of the virus in a plasmid, that is this thing, and then this is a genetic construct, and then they use this construct and they put it inside plant cells. This construct is translated is transcribed and translated as well. So we have the genome of the virus and we have the production of the proteins. Okay? But from here we have the production of the genomic transcript, so the genome of the virus. Okay, and this is a way to start the infection cycle or the infection of this virus because the positive sense genomes are produced in the cell. Here what they used is a technique that works with protoplasts. These are protoplasts is when you can take a tissue, you can grasp the tissue, you can isolate the cells, and then you break the tissue, and then you have the cells in a liquid medium without the row. This is what you see. So here, what they do also is in the genome of the virus, they introduced a reporter protein. A reporter protein is a protein that, a gene of a reporter protein is a gene that produces a protein that is fluorescent, and then you can see it in the microscope. Here you can see two colors. The green color is the amount of positive sense RNAs. And the red colors are chloroplasts of the cells, where the photosynthesis is performed. So these are some cells in which infection has started. Okay, in the first class, in the first session, I was saying that if we want to do a mathematical model, we can write a diagram. So this is what we did. This is a schematic diagram of the system that allowed us or facilitate us the construction of the model. Here we have different processes because, of course, we need to make a model that is based on the experiments. Here there is something that is not naturally occurring in a viral infection, but is this transcription. Because this is introduced in the cell, and this plasmid is continuously producing the genomic RNA, the positive sense RNA. We have a source of positive RNA, that is the complementary copy from the negative one. So here we have to take into account two processes for the production of positive sense RNA, and we will see it now. So here we have different processes. We have transcription. Constitutive transcription means that this RNA is constantly produced from this structure. Then we have the translation of the positive sense to synthesize the polymerase that will make copies from positive to negative and from negative to positive. Okay. Here we could do different models. And actually, in the references that I showed you, we have different models. Why? Because we have a lot of models here. We have this one, which is constant. We have the genomic RNAs, the antigenomic RNAs, the negative ones here, and we have the polymerase. So, we are interested in the quantification of the positive and negative strands. So we will not consider anything else than the genomes. Here you can see this P stands for positive sense RNA, so the genomic RNA here, and N for minus, the negative RNA. You can see here some familiar things. So phi is the famous logistic equation because we are assuming that the two strands are made of the same building blocks and they are competing for some resources. K is the carrying capacity. This is clear. This allows us to mount the system and introduce competition for resources. Then we have this degradation, spontaneous degradation of both genomic strands. Despite, as you will see, the kinetics is mainly amplification. So one might expect that these degradation rates would be low. And then we have three important things. So this tau, which participates as a constant in the production of the positive sense strands, what do you think comes from here? This tau, does not depend on P. It's just the constant production of P, which is this process here, transcription. And then we have, since we are doing complementary copies from positive, a positive one comes from a negative one, and a negative one comes from a positive one, here we have to change the variable. So for the production of positives, it will be proportional to the number of minus strands. R is the replication rate. And one important parameter is alpha. What is introducing alpha here? What is introducing alpha? So if alpha is equal to 1, it means that the production from positive to negative is the same as from negative to positive. Then I have geometric replication. This is a very easy or nice way to introduce this thing. But if alpha is positive but close to 0, then it means that the mean contribution will be from negative to positive, and we will have the stamping machine. And finally, after some discussions with the referee and with the editor of the paper that was in a conference today with us, they were complaining because they said that, and they were right, we didn't took it into account at the beginning, that when you have in the same cell, you have reduction of positive and negative, there may be some inhibition. And then including this term here, that let's say, we could find in many different ways, but we use just simple Michaelis method in which we have the inhibition of production by the other sequence. Okay, we have the model. Now, what we can do? So here I'm showing some experimental replicates. This is the data that we had. Actually, we had 3 independent replicates. This is usually what experimentalists do, usually 3 replicates, because the experiments are very costly and take a lot of time. And we have a time course, a system of protoplasts, identification in time, the positive sense in blue and negative sense in orange. And for each time point, we have 3 measures. In some places, we have only 2 or only 1 because, when we are doing experiments, there are a lot of issues. And I think this has said this times a lot. The first thing that we have to do when we need to fit a model to a data is to look at the data. This is the first thing that we have to do. So what would you say about this data? This is in linear log scale, right? So it grows until 10 to the power of 6, 10 to the power of 7, depending on the replicate. But here we can see, let's say, or I can see a couple of things. What can you see from this data? Anybody here? In relation with what we are trying to investigate, the mode of replication. What would you say here? If you had to bet some money, what would you say here? What kind of replication mode we may expect? These are the positive sense, let's say 10 to the 5 to 10 to the power of 6, and negatives 10 to the power of 5. Is this geometric replication? No, it's not geometric because we have a very large asymmetry. It means that we have more production of positive sense strands. And another important thing for the model. What kind of dynamics one can identify when looking at the data? Anybody? Do you have oscillations here? Do you have like steady increase, like a logistic function? It seems that you have a kind of increase and saturation. Okay. I mean, even the replicates are very similar, and they are different experiments, and this is very good because sometimes you do an experiment and you have a time series, and you repeat the experiment and you have a completely different thing when you have a problem. But this is not the case here. So then our assumptions of the model, of course, I did the model after seeing the data, of course. And this is why we introduce the logistic term here. So we did not publish this in Science, okay? We published this in Genetics, but this is the main article that I was reading to prepare the fitting of this system. And it's an old paper that talks about optimization and simulated annealing. Okay? It's very interesting paper because here they do optimization for computer architectures. So it's a very innovative work for this year. So what we say is that these points here, these experimental points are we denote them as y sub I. So y is the experimental value at time I. Then we have, of course, we have our dynamical model, which is this one, and we have the will species. Okay, this is the vector of parameters of the model that we have. Here we will not include the initial condition in the parameter. If you remember, Luis was saying that many times we need to include the initial condition because when we have exponential growth, this is very sensitive. Why do we not include? I'm asking because I don't want you to get asleep. Okay? So why I did not include here the initial condition? What is the initial condition? What is the initial condition here? Zero. I have never been positive because the system starts when this parameter here now starts accumulating P. And this is just a matter of the experiment because in order to start the infection, they need to include this construct that the initial condition is zero. The system by itself, without is positive, just starts. So I don't care about the initial condition. Then here, because these models usually behave very well, there are smooth, there are non-stiff. I use a four-order Regecuta with a constant time step size, 0.01. And of course, here I define the time scale in hours, so 2 hours. And then I just iterate it by 8,000 times to get 80 hours. This is the time scale of the experiment. Then the solutions of the array, of these two arrays of numerically, these are 5K. That we get a solution, xi, so we get a solution for xp and xn, given a set, or given a set of these by different parameters. So this is something that we have always to do when we need to feed data to a model. So here, what I used is least squares computed from the residuals. Okay, this is what we call the objective function. This is a measure of the distance or the error, let's say distance better, between the simulated data, simulated here with the arrays, and the experimental data. But here you can use the L2 norm, or you can use other norms. Of course, here I do not have single time series. And I do not have a point for every time series. I have different points, and I have two variables. So what I want to do is to make simultaneous optimization of SP and SM. And here what I use is function that is called, this is called a Pareto function. And what I have is that I have the objective function, it's lambda. The first one was SP plus 1 minus lambda Sm. If I set lambda to 1/2, then I am giving the same importance to the two data sets. And this is what we did here. Of course, here you have theorem. And here we fixed lambda to 1/2. For example, imagine that you have much more points for this one, the SP, that for the SM. Then it is better to weight the fitting with a curve that you have more data, you have more points. So here we have this objective function that it's the least squares of the residuals that are computed simultaneously with this Pareto function, and then we applied the simulated annealing. So the first thing I did is like to look for a seat for the simulated annealing, and I did it by brute force. So I made the Monte Carlo simulation, a lot of parameters and a lot of times to get seat. Then, as Luis explained yesterday, when you have the simulated annealing, what you are doing is that you are exploring a region in the fitness landscape very in a very large detail. So you have exploitation, and you have also exploration, because some parameter combinations that are not optimal can be chosen, especially at the beginning when the annealing is low, when the temperature is large. So what we do usually is we make small random modifications of the seed, then we obtain the solution of the OU with this numerical method. This is a very standard simulated annealing thing. Then we build a new objective function as mu, and we build from this Pareto function where SP and Sn are given by this distance measure. And then if the new parameters improve, that is, the S mu is lower or equal than S, then S mu is accepted, and S becomes S mu, and if it's not improved, otherwise, the new set of parameters is accepted with this probability, where the capital delta S is the difference in fitness, and this is exponential, and T decreases exponentially a long time. So what we did is we iterated this algorithm 10 to the power of 5 times, and we did it 500 times. One important thing I have to say is that this solution here, this can be a solution of a system of differential equations, but it can be a solution of a cellular automata, or it can be a solution of a PDE system, or it can be a solution of a stochastic simulation, an LSP. Okay, so this is what we call the simulated data, but it can be any kind of model that we have been seeing in the sessions. After we have the results, these are 3 replicates. The diamonds are the positive sense strands, and the squares are the negative sense strands, genomes, and the experimental data. And the solid lines are the best fit obtained with the with the model I showed you. So here I have to mention several things. So you will see, for example, that here the fitting is very good, or is good, and here the fitting is good, but here mainly it's not very good. Here more or less it's good. Here it's good, here the beginning. And here we have also some discrepancies here. And this is in log scale, so the differences are quite large. This is a system of overviews, deterministic. We are not considering noise, which might be very relevant here at the beginning, because we have low number of genomes, and here noise would be expected to be large. And I say that this figure is good because we are really interested in, let's say, in other words, our model makes more sense for this region here than here, because our model is not considering noise. Okay. If you have any question, just ask. Yes.
If you try to change the parameter lambda in the Pareto function to see if it changes a lot, like maybe you are giving much more importance to the class branch, like if you get some fitting or
yeah, we didn't do it, but I think the differences may not be very important in terms of the values of the parameters. But it is true that, for example, here the data for the negative sense strands, here we have no data. But at the end, for example, this shape here largely depends on this process here, assuming that this is exponential growth, we have competition. but this is a good point. I think it will not change a lot, but it could serve or it could be used, for example, to compensate these things. Because here, for example, if we give more weight to this one, probably this fitting would be better. Yeah.
Your target function, you don't normalize the data points to some mean value or some typical scale, but your data points are on different scales, right? Because you have it on a log scale. So if you have a difference of the prediction versus the data point for the minus sense, negative sense, it does not really increase your target function a lot, while there's a big impact of the blue points.
Yeah, actually, in the way we normalized the data.
Okay.
Yeah, but we are showing it like per scale.
Okay.
Yeah, good point. Yeah.
And what was the reason in this research paper that you chose this method? Because I could think that there are probably many different ways to fit these. You could also do a queries or something like that.
Yes. I use this method because it was the one I know at the time. My office partner had to work with this method, and he explained me the method. Yeah, but now probably I would use the deterministic approach, because the system behaves very well, or the macroevolutionary algorithm that I will explain later. This is something I have been always aware of, and this is something that I said, okay, when I have time, I will try to do a macroevolution for this data, but I don't have time.
Yeah.
Yes.
Short last question. Why did you fit one data set per replicate? So I would have, just naively, I would think you would fit one data set for all three replicates.
Yeah.
In the counted variant.
Yes. I didn't say that here. We always have the same, or I always have the same doubt. Should I make a mean and use the mean values, or should I make the fitting like 3 times the point? Here we need the second one, and I don't know if it was clear from the formulas I put it here, because this was like a general thing. So here. But we did it for the 3. Okay. It's in the paper. It's on paper, and there are some things that I don't remember. So these are the results for the 3 replicates. These are the medians of the 500 realizations for the parameters. Here there are some interesting things. So the first one is that it's alpha. This was the goal of our research, to determine alpha. This is like, if you multiply by 100, this is like the percentage of genomes that are replicated from positive to negative. So since this is, it means that the 6% of the times that the genome is copied from positive to negative, it's a stamping machine. So if this number would be 1, then we would have geometric replication, but it's very small. It means that the contribution is mainly from negative to positive. It's a stamping machine. In one replicate, we get like 0.06, in the other 0.065, and this one was like a little bit more large, probably because of this problem here. Okay. Then the replication rates were quite similar. I mean, the numbers were very similar. Then the derivation of molecules of genomes per hour is usually low. I mean, here we have dominance of production in contrast with derivation. The carrying capacity, for example, here what I did, just by looking to the data, I made the system to be able to explore between 10 to the power of 4 and 10 to the power of 7, I think. So, and here, of course, I would assume that the system has achieved the equilibrium, let's say, and probably you have to tune it a lot of times in the experiments, but sometimes if you leave this more time, it starts degrading because the cell is accumulating substances. And here we stop it here at this stable phase. Then we have this tau, which is the constitutive production of the positive genomes that was quite small also. And this is the interference coefficient that we have from interference between the strands, which is also very small, and the numbers are consistent. These are the replicates, and these are like the average values. It's not the mean, it's the median, I think. Okay, and the alpha was like 0.074. So it means that approximately 93% of the replication is stamping machine. And this was the first, let's say, direct evidence of this replication mode for a virus. I mean, direct because we quantify directly from the production of genomes. Some years later, there were some other groups that did for polio, I think, using also the genomic mechanisms of the genomes. Okay, so now I will explain you some results that are more related with the dynamics of the system. Of course, we need the experimental one because we are interested in the behavior of the system. Let's say in a natural infection, we just cause our model is not modeling. We are just using a very simple model, abstract model, to see what kind of dynamics one may expect from these different modes of replication or these alternative modes of replication. We remember, and I don't know if it was in the first session, I said to you that we can model a system by different ways. And for example, here we will see that what we did was just, this is the model that we used to feed the data, but in a natural infection, one may not expect now. Now would be zero in a natural infection. And also, from the experiments, we quantified psi, I don't know how to say, psi, was very low. So what we did is, what happens if we study the system as, let's say, logistic equation, but of course, the difference here is that we have complementary replication. Okay, it's not producing P. This will be a logistic equation. M is producing P, and the other way around. And the simple model, the dynamics is very simple. That tells us interesting things. So these are the fixed points I will show you later. But here there is a bifurcation parameter or a bifurcation expression that depends on the model parameter. So this is I would say mode of replication, but it's not the mode of replication. It's the asymmetry within the production. This is the decay of the sequences, and this is the replication rate. And this is a critical value that is involved in our bifurcation. We have a logistic system with degradation. We have a transcritical bifurcation. And this is what we see here. This is for delta. When we increase delta, fixing these values to these numbers, that are the numbers that we got with the experiment, we can see that when delta is smaller than delta C, which is r square root of alpha, we have coexistence of the positive and negative. Here you have to realize that alpha is very small, so we will have much more production of plus strands. And this is the bifurcation diagram. This is the concentration at equilibrium. This is the phase portrait when we have the origin, which is unstable. And then we have this interior equilibrium, which is locally synthetically stable. Well, actually, this is globally synthetically stable because it's the only term that we have here. When we increase delta, this point travels along this manifold or this place here, collides here, and there is a change of stability. Okay, so then the origin becomes globally synthetically stable. This is very trivial. But one interesting thing is that If we make a diagram, 3D bifurcation diagram, tuning alpha from stamping machine to geometric replication, and here we put delta, we see that when we have replication that is close to stamping machine, this critical value is lower. It makes sense. It makes sense because we have the negative sense strand that is producing a lot of positive sense strand, and we have few negative ones, and if these negative ones are removed, then the system stops. But if we have production of both of them, the system is more, let's say, resilient, or it's more robust. Okay. This is the example of the quantitative modeling. That because I showed you before, if anybody online has some questions, just turn your microphone. Okay. In the other articles that we are developing, we were very interested in how the mode of replication impact on the accumulation of mutations, and specifically how the replication mode is influencing the error threshold, the error catastrophe, virus extinction, and also other, let's say, virologically meaningful processes. And this is a different course. So if anybody is interested, I can provide the references. Okay. So now I will talk about a heuristic method for optimization that I have used 3 or 4 times in my research. And I have to say that it works very well. It's easy to program. Depending on the data you have and the model you have, then you have to stay a lot of time programming it, but it works quite well. This is the article that I was explaining to you. So this method was conceived by Jesus Marina and Ricard Soler. Ricard Soler was my PhD supervisor. This was in 1999, so I did my PhD later. And this algorithm is based on the works that Ricard Soler was doing in Macroevolution and outcome dynamics. What I will do is to explain the advantages or the differences between the macroevolutionary algorithm. As I told you, this method I've seen very, very few times in the literature. And I think it's worth to explain it because it works very well. What I will do is to compare a little bit these algorithms with genetic algorithms. And next week, Luis will provide a very wide explanation about genetic algorithms, which are very used. And then I will give you an example, let's say a general view of these algorithms, a general example, although sometimes they are a little bit contradictory, but you will see. And then I will show you a specific example. Why? Because these methods are not fixed, are not static. I mean, there is not a single way to do it. I can do it in many ways. The idea, or the point is that you touch the idea. And also, I can adapt the search methodology to the problem I am addressing. So it depends largely on the data, and especially on the landscape, that sometimes we don't know it. So all these techniques come from a subject that is very, very exciting, that is evolutionary computation. And mainly evolutionary computation refers to computer techniques or software or processes, algorithms, computer algorithms that are inspired by biological mechanisms. Why? Because biology is very wise. A lot of thousands, millions of years of evolution, of tuning, of adaptation, and we can do it very fast with a computer. We don't need to wait 3,000 million years. So usually, evolutionary search, or what we call heuristic optimization methods must find minima in a river landscape, in a very complex landscape. So you have to imagine this landscape as these are some parameters, for example, and this is the color is the fitness that the use of these parameters give to explain the data. So if we twist this image, this would be the local minima that we are trying to find. Of course, here you can see or you can differentiate that this one probably is the best solution, but you have a lot of local minima. Then, if we have a landscape like this, we will be in danger to be trapped in a local minima and not finding the optimum. So, for the canonical genetic algorithms, as we will see the next week, we have like a population of n individuals which are described by bit sequences, and this is very important because we are working with bits. Of course, we have a populational generation n that evolves in some way of changes to the population of generation n plus 1. And here we have what we call the operators. This is the genetic algorithms. You will see that in the macroevolutionary algorithm is not so different, but it's very different. So we have, typically we have the selection operator, the crossover operator, that is we have the combination between the strings that are encoding some parametric information and the mutation operator. Mutation operator allows for the exploitation of precise solutions, and crossover operator allows to jump like at larger genetic distances. And this is what happens in biology. Sexual reproduction, the dimension of sexual reproduction is for this reason, the crossover operation. So genetic algorithms are characterized by simplicity and flexibility, and they can be applied to combinatorial optimization programs with a large space of candidate solutions. When we have a landscape that is very irregular, we have a lot of local minima. And these are the operators. So the genetic algorithms are based on the Darwinian metaphor. So we have short-term evolution of the parameters. We call microevolution. This is a good way of doing things, and biology works like this. But in biology, we have different scales. So what Ricard Soler and Jesus Marín proposed at the time, and I think it was a very important contribution in the evolutionary computation area, is that we can consider a different time scale, the macroevolutionary time scale. And it means that we will have extinction or macro extinctions and diversification of species through their interactions. When I talk about species, I'm talking about vectors of parameters. So I have a system with 5 parameters, and I have a family of vectors with different values. These are the species. And as you know, in macroevolution, there have been macro extinctions in different geologic areas. For different reasons. For example, in the Cretaceous, in 16.5 million years ago, we had this huge impact of a meteorite in the Gulf of Mexico. And this is one theory, and this is a theory I believe, nothing about aliens or things like this, because there is a tremendous hole in the Gulf of Mexico, and this is because of the impact of a comet, of a meteorite, and this completely changed the conditions of the planet. And a lot of species disappeared, and there have been a lot of mass extinctions. In the fossil record, there are a lot of different mass extinctions. Some of them already removed 95% of the species in the world. But I'm not an expert in these things, well, in anything. So we can have large extinctions, and this means that this can generate current population responses that are really different from the slow Darwinian dynamics that we have with the genetic algorithms. And this is the key. This is I think what makes macroevolutionary algorithms sometimes, not always, a better approach to the genetic algorithms. Sometimes, not always. The article by Jesús Marina Becar Soler took different landscapes and compared the performance of genetic algorithms with macroevolutionary algorithms. And in some of them, they saw that macroevolutionary algorithms converge more rapidly than the genetic algorithms. But at the end, the genetic algorithms also was able to find the let's say, the good minimum. So this algorithm is based on biological models of macroevolution. This is one example published in Physical Review about self-organized criticality and extinction in models of large-scale evolution. But there are others. I will tell you later. And here what we consider is like an ecosystem where the dynamics of our species, that are our combination of parameters, are based on ecological relations. Now I am explaining it in a general way. Probably it will be difficult to understand, but then I will show you an example because we can do it with many different ways. And I will provide you a very simple way to do that works very well. So the links between these species are very important to decide if they survive or they do not survive at each generation. So this is like a genetic algorithm. We have a population of species or vectors of parameters, discrete, and then we iterate. And we apply some evolutionary rules to this population that in case of genetic algorithms are Darwinian dynamics. So we define the state of a species I at generation T as SIT, that is, can be 1 or 0, alive or dead. And here, let's assume that P is constant, so we have a constant population of parameters. We could play with this, we could make it not be constant, but we usually assume that it's constant. So the relation between the species is represented by a connectivity matrix, this WAG, that changes in time, and measures the influence of a species I with J. So, for example, if I have a trophic network, I can have an adjacency matrix where I have the interactions between all the pairs of a species. It can be positive or negative. I can have cooperation, or I can have competition, or I can have cooperation or cooperation. And I can give a value depending on this interaction between species A and J in an interval from -1 to +1. I can use different numbers. So at the end of each generation, and this is important, all species that have died are replaced by others. And these ones can be completely new species because they come from outside the ecosystem, or they can be the existing ones with some mutations, with some changes. So in this sense here, we are considering the mutation operator, but not the recombination one. We could also do. I don't know if anybody has done it, but we could also do. So each generation in the biological model, this is like the biological model, consists of different steps. So we have random variation. So for each species I, a connection WAJ is chosen randomly, and a random value between -1 and 1 is assigned. Okay, this is like the variation in the interactions of the species. Then we have extinction. So we have this value H that is like the contribution of all the interactions of all the species with it. And at the next time generation, the selected species I will survive if this function HA, which is a survival coefficient, is larger or equal than 0. And if not, it will go to extinction because the inputs will be not positive. And this step allows us for having extinction of the species. And then we have the diversification, because when you know that when there is, for example, you have a forest and there is a fire and you have a macro extinction, then you can have movement of new individuals and you can have procedure of diversification. But it's mainly that in the vacant sites that have been removed because they have died, we are introducing some variation. And this is like the mutation operator. This is a very abstract ecological jargon. So this model, as Soler and Susana Andrea shown here in this article and in the Nature paper 1997, with this kind of dynamics or model, they were able to reproduce macroevolutionary data, fossil data. So these biological models reproduce the statistical patterns observed in macroevolutionary data. It is important to say that in this model we are decoupling macroevolution from macroevolution, or in some sense. We have these mass extinctions, which will allow our system to be very exploratory. Because genetic algorithms usually involve selection and continuous adaptation, despite you can have jumps. But this one allows for massive extinctions. Okay. Now I will go on with this general explanation, just to be more precise. So the macroevolutionary algorithm, this should be an E, we define the d-dimensional fitness function f as the multidimensional function that we want to optimize. So here we are optimizing with, let's say, a large amount of parameters. Our goal is to find the best values for this d-dimensional vector to our problem. And our species, Si, are given by a vector of parameters that belong to a subspace, which is d-dimensional, where V is the number of parameters. And P will be a good approximation if for Q we have this fitness function of Q lower or equal than the fitness of P plus some error that is like an error threshold, and where P and Q are different individuals. This is not important. As with genetic algorithms, in macroevolutionary algorithm, we start with a population of P individuals or N individuals, a number of individuals. And also, evolution in time is by successive steps of given operators, and we iterate this system. So, the idea, as I said, is that the macroevolutionary algorithm will choose through network interactions which individuals will be eliminated or not. This is to guarantee exploration by new individuals and exploitation for better solutions that we could find that perhaps we have a close solution to this good one that is better. Then it's essential to establish correctly this relationship between individuals. In the example I will explain you, this is done, let's say, like in a mean way of the population. We will not go individual per individual because this is also very costly computational. Okay, this is from the article. I will go fast through this because it's more or less what I have explained. So the idea is that we are considering these ecological relations in terms of fitness to decide if they survive or not. And for example, if we have two close solutions, although this sometimes we don't make to do, we have like this expression here that gives this function or this connection matrix between the A and J is given like this difference in the fitness. Okay, but this is the same as I repeated. So this is the idea of the biological models of evolution translated to the macroevolutionary algorithm. And this is important. We need selection operators. And I already explained it. So it allows us to decide which ones will survive or not. And for doing so, we can do it by different ways. And we will show it in another one. Okay, colonization operator. This is when we introduce this small variation. And here, for example, there is an important thing is that the macroevolutionary algorithm, you can also do annealing. You can do annealing. What does it mean annealing? It means that at the beginning, the system is temperature is very large, and you have a lot of noise. You have fluctuations that can make the solutions to jump randomly. As you compute with solutions, in principle, you are like getting close to other places, and for you it's not interesting to jump a lot. So you want to keep still there, but still having some chances to jump. And when temperature cools and goes down at the end of the simulation, then it means that you are not allowed to jump so far because in principle you are in the correct region. Okay, this is the idea of annealing, and you can also do it here because here we have this temperature thing. Okay. Let's do like a diagram of a particular macroevolutionary algorithm example. So here, for example, we define first, this is like how we should do it. First, we have to define the population structure. So we have to choose a size of the population and the vector composition P. For example, 3 parameters. We have 3 parameters. Then we have to define the ranges for searching the parameters. And this is sometimes very difficult. And this was explaining in one of his sessions that he was like, by means of analytical and computational methods to reduce the space of search to a compact space by means of different techniques. And it depends on the problem. For example, in the example I will show you later, the parameters were easy to define the ranges. You will see why. Then we have to initialize the whole population with random numbers, the ranges that we have defined with a uniform distribution. From a more practical point of view, usually the view is build a matrix in your computer. You build a matrix when you have the rows are the different species, E is the size of the population, and then each of the species is a vector of 3 parameters. So P01, P02, P03, and these are the vectors of parameters. And then you can do it as you want. I draw in C, and what I do is I add a column, and here I put the thickness values. For example, if we choose a size of 12,000 species, we can consider, or what I do is I consider 3 blocks. So n divided by 3, which I call the best species block. Then the other n divided 3 plus n divided 3, the colonization variance that I built is the best species block. And then the n divided by 3 empty sites, what I call macro extinctions. But you can do it whenever you want. You can here, for example, use 10,000, 1,000, 1,000. This is up to you. Depending on the methods, you will be more exploratory or more exploratory. Okay. And of course, at generation 0, we put random parameters, random values for the ranges of each of the 3 parameters that we have. So now, this is what we need to do at the beginning. We iterate the or we start with the algorithm. So the first thing that we have to do is for each species, using these parameters, compute the fitness. And usually what we do is we take a zero, we take these parameters, we put these parameters in a mathematical model or in a simulation model, and we compare the solution obtained with the model with the data. And we provide a measure of the distance of the fitness. And we can do it with the least squares, with the L2, or with any norm that we want. For example, as I was saying before, we can do it numerically or using simulations. For example, I have an article that was an idea because I had to implement, and I know in the good sense, I had to implement a bit string model to reproduce experimental data. And these were stochastic simulations because I was iterating the bit string with fitness landscapes, whatever, different processes, and then I had to program a macroevolutionary algorithm to fit the probabilities of the Monte Carlo model to fit a particular type of plot. So I spent a lot of hours and a lot of coffee. So imagine for the first species, we have a fitness 10.5. Okay, then we repeat the process. Here are random parameters. And at the end, we finish with this set of fitness for each parameter combination. Okay, what I do then? Here is where I compute this matrix of interactions by globally. And what I do here is I compute the mean fitness of all the populations, because I need to start with something. This mean fitness is 7.66666. Okay, so at generation 1, what I do is selection, variation, and macro extinctions. So what I do is I select those species with fitness smaller than the mean fitness. And I put all these candidates the first block in the DSD block. Here I have 2. I have 1.5 and 4.5. These are the only ones that are below this mean fitness. The other are above. So these ones, I kill them. So I put this one. Sorry, I have to say something. This is an example for 3 equals 6. Okay? This is a very simple example where I have a population with 6 species, only for you to know. And I do 3 logs of 2. Okay. So I select this species with these characteristics and I put it here. But now the one that was coming from S3, P3, 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 1 to 3, for 5 here, it's S0 in the generation. And the same for the other one. This is S5, but here is S1. And now I have 6 empty sites because all the other species have disappeared, there has been a macro extinction. So here what I do is I take this block here in this blue, light blue, I take the values that I have selected and I copy them as 0, I copy them to S2 with some variation. And then I will have another fitness. But in principle, will not be very different from this one, but we don't know. And the same for the other one. And the other empty sites, I put random numbers from the initial ranges, completely random species. They are coming completely random species. And this is extinctions plus colonization with I introduce the rest species with variations, and then macro extinctions where I introduce completely random parameters. And then I end up with this table here, and I compute all the things again. First one 4.5, 1.5, and then the others, the value I get. also comparing the simulated data with the experimental data. And now, here, I have random values. I can have a very low fitness or a very large fitness, but usually the fitness is not good. And now what I will do is to work with the mean fitness of these two blocks. If I want to be very restrictive, I will only work with this fitness, with the fitness of the best solutions. But I can do also with these ones. So now I compute the mean fitness from good solutions, and I will do from now on. Generation 2. What I've done here is the mean fitness was 3.05. So at generation 2, I select those values with fitness smaller than 305, 08, and 15. And one thing that I also do, I will show you, is to place the values here from low to high fitness. So I order the values from low to high fitness. In the sense that I always have the better solution in the first place, and you will see why I'm doing it. And here I do the same. Then I take these values, that were placed in this block, and I make some random mutations, and I will get another fitness, and then completely random parameters. And I iterate, repeat. So at the end, what I have is a population optimized in principle, have an optimized population. These two blocks, this and this, and especially this one. So this is the block that I will probably use, the first one, that, I don't know, it can have 5,000 individuals. I can use 5,000, 5,000, 5,000, or 5,000, 2,000, 3,000. It doesn't matter. It matters, but it's not important. So I arrived to an optimized family of parameters, the USB block. Something that I usually do when I'm doing the simulation, I am always, well, it's here in the first row, that I am recording the best parameter. Always. This is what is called tournament selection. You will see that I will use jargon from genetic algorithms. An important thing is that, not in the case I will show you because we have few parameters, but when you have a lot of parameters, you may obtain two good fitness with very different parameters. And then you have to check for correlations. I mean, perhaps you have a process of growth and decay, and you have like 10 growth, 1 decay is okay, and 30 growth, 3 decay is okay. Then you have to work with a ratio, so with a relation. But this is a very hard thing to do. So what we can do, here we have a lot of data. We can do averages and variance studies of these good candidates. And we can see the dispersion. If dispersion is very small, it will mean that we are in a place of the space that is good. Can repeat this process. So here I will have, for example, 5,000 good candidates. I can take the best one and I can make the averages between these ones, then between all these twos and C. Another thing that I can do is I can make this process a lot of times. I can make replicates of this process, and then I will have 1,000 or 100 times 5,000 families of parameters. And I will explain a very large parameter space or a lot of times. We can also track the mean fitness of the best species in this block, for example, along the generations of the algorithms. We can also follow the distance, how this error changes in time, because it's very useful to visualize how this performance is done, how this optimization is done. This is something I highly recommend. Now I will end with an example in which I use this macroevolutionary algorithm for a specific problem. This is a work that we did in collaboration with Cristina Alcaide and Pedro Gomez from Murcia. They are plant biologists. And here they collaborate with me and with Santiago Lena because they had some nice data. This is the work I will present, and I will explain you a very funny story. So what do we do? The way we invested what is the effect of temperature that mix of single and mixed infections in plants. Okay. So here they had two viral strains, two different viral strains that infect tomato plants. One is called CH2 and the other is EU. This is from Chile and this is from Europe. And each viral strain has different effects on the plant, on the tomato plants or family of tomato plants. This figure is a little bit complicated, but here the idea is that you have so they inoculate the virus alone, CH2 in plants, and follow the accumulation. Then they use new strain and follow the accumulation, and then they mix the two viruses. They did it for two different temperatures, at 20 degrees and 30 degrees. So here we have population dynamics, temperature. These are the main processes. So here you have in black the single infections. This is data 7 DPI. This is days post-infection. So this is time, 1 week, and 60 days post-infection. So this is CH2 in black for single infection for CH2 at 7 days after starting the experiment and 60 days. And this is the same for the mixed infection. Okay, and the same for 30 degrees. So here we have kind of time series. But we have 3 points. And this is probably the only paper that you will find with a fitting with 3 points. But you will see, it makes sense. Okay. A very fun thing, I hope the referee of the paper is not attending to the course, is that we put all the things in this paper here. Evolution, very nice journal, Oxford Journal, Evolution, very nice. And we put here the mathematical model, the fitting. And one of the referees said, this is very nice if you remove all the theoretical and computational part. And then the paper will be published. This is what they said to us. And I said, okay, so let's do 2 papers. And we did 2 papers. Probably this guy was not, this referee was not used to see the equations, but the equations are very easy, you'll see. So we have this data, and we want to in some way estimate parameters, but here the most important thing that we were interested is the effect of temperature on the competition of the viruses. And this is why we have the experiments with the single infections and the mixed infections. You can imagine that these experiments are not easy to obtain data. This is why we have very few data, because this is 7 days post-infection, 60 days post-infection. They have to smash the plants, calculate the viral load, and it's impossible to have a regular time series. It's very costly. This is why we have 3 time points. 3, but with quality. Modeling, okay. So how can we introduce temperature into evolution dynamics? We have to invent some function. Well, it's not invented because it was published in another work, but we found this equation here. Now I is the strain EU or CH2, okay? So these are the variables, the 2 viral strains. And R of T, here T is temperature, and R is the replication rate of the virus. So we have this function. So we have that depends on T. With this temperature, we need to define a minimum T and a maximum T, and then we have these two parameters A and B. Mainly A determines the height of this function, the magnitude of the temperature in the replication, and B is how symmetric is this function. These are some examples in which we have these are arbitrary values for different values of A and B. We have different shapes, and maybe what we have is like a shape that we might expect that systems behave like, that is at low temperatures they grow slowly because these are plants, these are per kilotherms, so cold, of course, plants do not have it, but they entirely depend on the temperature for growing. There is like an increase with temperature with a maximum, but usually it's for bacteria, for example, it's 37 degrees, and then we have a decrease. I think the battery is no, you cannot see the pointer. You can see the pointer right here, yeah. So and then we have the decrease. Okay, so now we can introduce the temperature in the system. Let's go for the single infections. Here also we have a look to the data, which was very limited, but our intuition told us that it followed logistic growth. And what we did is just to use the logistic equation, where we have different experiments for strain U and strain CH2. Okay, sorry. For the mixed infections, we use a similar model. This is a local alternate competition model where the species can compete between them in this system here. So this is intraspecific competition, and this is interspecific competition. And the C is the strength of competition that C exerts on E, because they are competing for resources. Here, we are like doing in plant model. It's like we are considering the whole plant. This is a very strong assumption, but this is reality. So for the single infections, it is very easy. This is a logistic equation in continuous time. We have two equilibrium points, the origin and K, which is the current capacity, and actually we can have the solution. We can integrate the system and obtain a closed solution. That is this expression here. This is the derivative of the function with respect to x, and it is very easy to show that, of course, here that depends on t is a function of t that is a constant, it's positive, and then P1, that is the origin, is unstable, and P2 is an attractor, where the system will go. Okay, and this is just for you to show you that the dynamics is very simple. We have one-dimensional phase space. This is the origin, which is unstable, and this is P2, which is K, and this is a potential function. This is a potential function that has different weights. I'm showing it because you can see that in the fixed points, it has nothing to do with temperature. It's not appearing temperature. It doesn't depend on the temperature of the equilibrium point. But temperature has an effect on how many times I take to go to a solution. Because time is in this, well, I would say multiplier. It's like when you do a stability analysis, here, if depending on R, I mean, if depending on R, you will go faster or slower to the equilibrium. Okay, and this you can see also with the potential. If the system goes very fast to the P2, then the potential will be very, very large. And for the two-species system, here it's a little bit more complicated, but it's very easy. We have 4 equilibrium points. We have the origin. We have these 2 points that the ones that represent competitive exclusion. Here, the first virus E wins, and here the second virus wins. And these points are in the invariant range of the free space, because one coordinate is 0. And then we have a coexistence equilibrium where the 2 species have positive solution. And these values do not depend on temperature. I mean, they do not depend on R. But as in the previous system for single infections, temperature has an effect on dynamics. Okay, in our experiments, what we saw, and I will show you very soon, we have coexistence of the 2 viruses upon time. So we are in a scenario where this fixed point, PEC, is stable. Because the system has an interior equilibrium, because we have the 2 populations. So I'm saying it because I could have taken a model, this model, and do a fitting. Okay. No problem. But if I study the model previously, I can understand the dynamics, and I can play and I can see if some parameters can have some values or not, if it makes sense. Because now I will show you that, for example, beta EC has to be lower than 1. If beta EC or beta C is higher than 1, this point here is not stable. And then in my range of parameters, I will fix from 0 to 1. And then I have reduced the search space. So this is the linearization of the system. This is the Jacobian matrix. Of course, for the 0, 0, if I compute the values at this point, so local stability, what I get is 0, 0, this cancels, and I obtain RET 0, 0, RCT. And RET and RCT are positive. So the 0 is a repeller. If I compute the values for E and PC, they are very similar, and one of them is negative always, and the other one depends on this relation. And this is why if beta C is higher than 1, one of these points is stable. And the dynamics go to PP or PC. It cannot go for coexistence. Then I don't have to search in the parameter space when beta C or beta C is larger than 1. Because I will not get to what I'm looking at the results. Of course, this is a model. The results are the reality. There are other assumptions. This has to be clear. These are the values for the other fixed point, for the interior fixed point, which is very complicated. But what I show you here is bifurcation diagram moving beta EC. You see that when beta EC is between 0 and 1, we have coexistence. So this is the numerical value computed, numerically with our integrator method. And these are eigenvalues. You see that under this range, the eigenvalues of the point PEC are negative. One is constant, and the other one is negative. So here, this point is an attractor. This is why I found it numerically here. But at this point, the EC, there is an exchange of stability because this point collides with PC. These are the eigenvalues of PC. And here you have the phase portraits. This is the case where beta EC is smaller than 1, and I have that these 2 points here are saddles, and I go to this stable node. And this is when beta EC is bigger than 1. And you go here. Okay. Usually what you have is that this point moves towards this point or towards this point, and you have a transcritical bifurcation. Here you have the other case here. So I have studied this model. Now I will explain all the steps that we did to apply a macroevolutionary algorithm to the data that we will see in few minutes. So here we define a population omega n with n vectors. So this is the P, the previous P, where the vectors are given by the two values of the function for the replication that make this shape, this relation between replication and temperature, the competition coefficients and the carrying capacity, and K is the individual of the population from 0 to n. Of course, here for single infections, we consider that this beta ij with I different j, from j and ij belong EC are 0 because we don't have interspecific competition. Here we define exploratory ranges. Beta ij has to be between 0 and 1. If not, we cannot have coexistence. It's impossible in this model. And AI and BI, you can see a very wide range. This is how we usually do it. You try to choose a very wide range. And K, this is mainly inspired by data, from 10 to the power of 3 to 10 to the power of 11. This is our exploratory range. Of course, here we have a lot of orders of magnitude between the boundaries of this system, and here we have also like 8 orders of magnitude, and here what we have to do is to sample the exponents of this expression. Okay, so instead of computing a number between 1,000 and 10 to the power of 11, we make a sample, a random sample in a uniform distribution of the exponents in order to be sure that we are exploring everything. And this is what I talk about coexistence. Okay. First, we initialize the population of vectors at time tau equals 0 with random values between these ranges here. And then at each time generation, we do the following. I'm doing the same that I previously explained. So for each vector, we compute the distance between the simulated data and the experimental data, now called epsilon, by means of least squares. So here we also have, so we have 3 time points in days, but for each time point we have several replicates. So it's not so dramatic, the situation. So here what we did is to adjust or to compute the distance, taking into account the different replicates with the residuals of, and this is like a normalization term, and F is a constant that we put because we got very large numbers. Okay, here we could use another norm, let's say L2 norm or whatever, but this F was, I think, was 10 to the power of 4, and it was to have not so large values of the F. And then here, I am doing the same that I explained for the simulated annealing. I am computing for the case of mixed infections, using a Pareto function with the distance for the two time series that are the same time point and have the two variables. This is only for the mixed infections. And then when I compute the fitness of all the vectors, I will have from lower fitness to larger fitness. From larger fitness to lower fitness. It means from less distance to larger distance. Because I assume that the higher the fitness, the lower the distance between the simulated and experimental data. I mean, if my species is close to the data, the fitness of the species is large. Then here I use, I think I use 10,000 individuals, no, 12,000 individuals. And what I do is I select a 25% of the individuals. Second. Yes, exactly. So when I have computed the fitness and the distances, I order them from low to larger distances. And then I select a 25% of the population. So the first n divided by 4 individuals. So the block, the BSB block, and I remove all the others. And then so at the generation t plus 1, what I do is I have these better solutions, these better species, this 25%, and I put them. Well, I actually have them in the population, and all the other sites are empty. So I am doing an elitist selection or truncated selection because I am selecting a percentage of the best ones. Then what I have to do is to fill the second block. And what I do is I select this 25% of better solutions that go from 1 to n divided by 4, and then from n divided by 4 to n divided by 2, I take the values and I modify a little bit. Okay? So what I do is I multiply everything, every parameter by introducing plus minus mu, and this mu is randomly chosen between 10 to the power minus 12 and 10 to the minus 2. So I'm making slight variations and I'm choosing it randomly. I do it this way. You could do another way. After we fill the third of the lattice, well, the numbers are later, it doesn't matter. With this variation of the best parameters, then I have like n divided by 2 empty spaces, which are filled again random. And then I return to 1. With this new population, I return to 1, and I do again the same, and I iterate. Okay, now the data. You will see that data is a little bit strange, but you will see that it makes sense because, for example, for the single infections, this is for the EU strain at 20 degrees and 30 degrees. And this is, of course, the initial condition that is fixed. And then we have these points at 7 days post-infection with some variation and at 60 days post-infection. Okay, and the simulation is done with a time scale of hours. Here you can see that the temperature makes a difference, okay, and this difference is not so large for the other strain. This work was done at the same time than the other one. In the other one, we did some statistics to see if these differences were significant or not. And here, what we estimate was this is the best parameter. The parameters with the best fitting. This is the first species of the population always. And we have the AI, VI, RI, and K. Here you can see why I fixed the carrying capacity between 10 to the power of 3 and 10 to the power of 11, because the system stabilizes at around 10 to the power of 6, 10 to the power of 7. So the system converges to these numbers here. So this is how these viruses behave as a function of temperature. Okay, single infections for one strain and the other. This is because I have very few points, and here I have 4 parameters, and I have replicates. Okay, but I have few points. So then, for the mixed infections, what I am assuming here is that the response of the growth of the virus to the different temperatures is the same, or more or less the same. It means that A and B will be the same for the same virus independently. And my assumption is that the changes that are now happening is because of competition between the two species. So here I have also the same, very few points, but here I am only estimating 3 parameters that are the betas and the K. Okay. The most interesting thing here is that for this particular case, that is just at 20 degrees, there is a switch. between the species. So at the beginning, it performs better, but then it decreases. And there is a switch. And the switch is important. I mean, here you have like one order of magnitude. And the first thing when I was working with this model was to ask myself, will my two-species model gather these dynamics, this change? Because this is, so the fitting is not very good because we have very few data. But is the model able to reproduce this switch? And the answer is yes. The answer is yes, because here you have the fitting of the model with a solid line. These are the experimental points. And here you have the data here. So we see that the competition at 20 degrees is much higher from C to E than from E to C. And this is why CH2 dominates. And here it's not so different. Okay, this is a particular example with very limited data. The message here I would say is that I will not believe these numbers a lot. But qualitatively, we have reproduced these dynamics. The important thing here is that I want to show you some outputs that when you program macroevolutionary algorithms, you usually should see or should observe. So this is how this distance, these least squares, performs along the macroevolutionary generations. For example, for 5 replicates overlapped for this virus at this temperature. And usually what you see is that you have a very fast decline of this distance, very fast, and then you get some kind of stabilization. And if you wait some more time, you can even see a larger jump. Okay. Here solutions are not here the fittings are quite good, but sometimes you need quite a lot of time to get good values. So this is a way we can visualize. Another way is to make a plot of that every time we have a best low distance, we plot it. And then this is not MA generations, it is improvement event. It means that we have these dynamics for the new emergence of a better solution than we had. And here you can see this is 10 to the power of 8, and you can see that it falls very rapidly in this scale, of course. And this is for 25 replicates, and you go to very low numbers. I mean, this method has a very fast convergence, and then probably because you are in a good region, you finally find, in principle, a good candidate. This is the same for the competition experiments. Here we have this DM distance, this composed distance between the two data dynamics. And here this is 10 to the power of 10, and this is almost 1. And you can see that in 10,000 generations, you have improved a lot. And just to finish, let me tell you the numbers I use here. I think I used 12,000 species, and then I divided the 3 logs in 4,000, 4,000, 4,000, and 4,000. It means BSV log, the data solutions, the variation, and the random. If I suspect that the landscape is very rugged or very complex, then perhaps I should make this macro extinction completely random new species longer. Okay, or I can change it and see how it modifies the dynamics. So this is everything for today. Sorry for this technical problem. If you have any questions, any comments, at the end you have to program it with your computer and iterate. This is what you need.
When you choose the variation of the best species, wouldn't it make sense to, for example, so not use a fixed noise, but
distribution?
Yeah. For example, it could fit two-dimensional Gaussian on the best.
Yes, you can do it perfectly. Here, the only thing I did here, you remember that this was talking about the anisotropic grid search, that you were not considering the same magnitude of changes depending on the parameters, because if you have a parameter between 0 and 5, it's not the same that between 0 and 1000. We did something similar, more homemade, that is to assume that this sometimes is intuition, that the magnitude of the change is like proportional to the number you have, but with this, let's say, range of variation, because actually, so this is mu. So if AI is 10, then I will have 10 plus minus 10 multiplied by this range. And this range was assumed to be uniform. So here I was sampling also the exponents. Here you could use a Gaussian, that actually you are putting it here, for example. I think perhaps this is too light. I mean, perhaps this is too much, because, but of course, if you have a number that is 10,000, you need here a very small number if you want to make a little movement around this place. And this is something that at that time for me was like, let's say, I will not say correct, but meaningful. But you can do whatever you want. Usually, when you sample ranges, usually very few times you have information that it's better to use a Gaussian distribution, for example. I don't have enough experience to comment about it. Perhaps you can ask Luis next day, because he has more experience. Okay.
That the randomly chosen species, they're chosen also from a uniform distribution.
Yes, here.
And really it makes sense to like promote somehow like the like really makes sense to maybe promote like less visited parameter sets or like areas of the parameter space or something.
Yes, but here it's not necessary, because I will be selecting these ones. So the idea is that I have the best candidates, then I have variation around, and I'm moving everywhere. And if in the second block or the third block I find a good solution, it goes immediately to the first block.
Yeah, but I think when you introduce new species, maybe it would be interesting to choose them from some spaces in the area in the parameter space that you haven't considered before.
I haven't.
You have.
Or I haven't.
No,
haven't. But I'm doing it in the third block. Yeah, it's very important. So here. So here I have the best. So this is a case for P6. So I have six species. So I make 3 blocks. 2, 2, 2. In the first block, I will put always the 2 best ones. In the second block, I will take these good ones and make small modifications. And in the last 2, completely random from the beginning of the range, from the initial range that I selected. And this is really very nice, because of course here it's very probable that I will get a good finish. But imagine that the mean is at the other side of the space. Then once the good solution enters here, it will go up here. This distance, this mean distance, this criterion is a way to do. So I can be more restrictive. And I can say, I will only accept those solutions that have distance 1.5, for example. Perhaps there are no ones that you have this distance. So it is also important to be aware of this magnitude of the distances. So the first thing that you have to do is to choose the objective function or the norm that you want to use, and see in the screen what are the numbers that you are getting. But here is very clear that, So for example, here, probably you will find a solution the chance that will be very good and you will have a very rapid decrease. And then probably here we will be like making this fine tuning with the second block and we will be enhancing a little bit but here you can see the jumps that you have in the best solution. So there are some like continuous changes and then you have a very sharp one. And this is probably because one of the completely random has colonized the population.
Your stop condition for the algorithm is always like repeating the process for the end generation.
Yes, this is a very good question. So here, it's very important to see the things, and here I can say that these are a lot of parameters. I mean, this is 10 to the power of 7 of a population of 15,000 or 20,000, and except the first block, the other two blocks are new parameters. So the first block is always keeping the parameters. This is a fitting with a deterministic system. The values will be the same. But when you have stochastic simulations, sometimes when you run a simulation with some parameters, you have a result. And then you repeat it and you have a different result. That will not be very different, but in principle will be different. But there are other criteria. For example, in genetic algorithms, in the work that we did with the seagulls, since Luis was exploring quite small parameter space because he made all these calculations and all these computations, he obtained convergence very fast. And what he saw it as a stop criterion is to say, if my solution has not improved in 200 generations, I stop. Here what I did is to live a very long time. Yeah, but the distance is so small here that probably a better solution would. So look here at the improvement event. It's very, very. Of course, you can say perhaps you have another solution that is better. This is something always that you can say. But and then of course you have to look to a fitting with your eyes, because I repeat, despite this very limited data, I was very surprised to see that the model, of course, here another point is that we didn't fix, so we didn't make the initial condition to change because we did some normalization, because we had some problems with the problems, meaning that we have to modify the data, not modify the data, but change how you put it here. And we did some way that the initial condition was 1, so we rescaled the data. And then this was fixed. Probably if we had left this free, the fitting would be better, because probably here you might expect that, for example, from this point, you go here, and here you should start bending. But if this, I will go here, and the model is very rigid. I mean, you could say, you have a fitting because you have some 6 time points and 3 parameters. Well, in principle, I would say there is no overfitting, but it's very rigid, the model. But this fitting is very good, especially for the black one. And you can see that the model perfectly reproduces this effect. And I was very surprised because I was not sure that the model that I was thinking was able to have this out competition in a transient time. Not at the equilibrium that we know, because we have done the equilibrium point and stability, and we know that playing with betas we can have a competition, or at least not a competition, but we switch here. And when I was doing it, I didn't have many faith, but it would not work, but finally it worked, and we got yeah. Okay. And there are comments about how to program it, and it depends a lot on the data and on the model that you have. If you have ADUs, it's very simple because you need the solver and the code, and you can, in some sense, make a general program or standardize. But when you are running simulations, well, you can also build, you can make separate simulations and then cooperate with the. But it also depends on the data you have and how you fit the data. If you make a mean, or you make simultaneous fitting with the 3 replicas separately, and then you join. So it's a lot of work. Yeah. But it works very well. This algorithm works very well. Okay, so thanks so much. I hope that you will have learned something. And if sometime you have to do a fitting, and especially if you have a lot of parameters, I would go for this method or genetic algorithms. And also, you have to be careful with these numbers, because you can have correlation between parameters, and then you can have very different parameters with similar results. This is something that you have. Of course, the more the parameters, the better the fitting. But for this article, I feel comfortable because at the end, it was like 3 parameters only. And the dynamics was very clear from the experiments. I mean, here there is a lot of noise, but again, we are more interested in this equilibrium, let's say, because this is like 60 days. The system has been a lot of time changing, and we assume that this is at equilibrium. Again, it's an assumption, but it's reasonable. And when we discussed with the experimentalists, they have like a sixth sense, they have intuition, and they said, yes, this is at equilibrium. Because we have been working and we have observed that plants from day 30 to 40, the virus is there, and the plant doesn't change. It's just there until the plant dies. Okay. Thank you.